Good afternoon and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO, and your host. Today, Dr. Brian Brennan will present Manipulating Memories to Help Treat Brain Illness. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 177 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such a great impact is because we do not limit ourselves to a focus on one illness or condition. Since 1987, the Foundation has awarded more than $440 million to fund more than 6,400 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, obsessive-compulsive disorder, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Brian Brennan. Dr. Brennan is Medical Director of the Obsessive-Compulsive Disorder Institute and Associate Director of Translational Neuroscience Research at McLean Hospital and Assistant Professor of Psychiatry at Harvard Medical School. He was a 2012 and 2008 Young Investigator. Our webinar will begin with Dr. Brennan's presentation, which will then be followed by Q&A. To submit your questions, please use the questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many as possible in the time allotted. And now I'm pleased to introduce Dr. Brennan. Brian, the floor is yours.
Thank you so much, Jeff. Really appreciate the introduction. Let me go ahead and share my slides, hopefully. Okay. So really thrilled to be here to talk to all of you today about obsessive-compulsive disorder and to talk a little bit about work we are doing specifically in the area of contamination-based OCD. So, even more specific. And I'll talk a little bit today about some of the background leading us to the project that we're currently involved with. So just to start off and give a little background, we are in the OCD research field. We are very lucky to have had several decades of exceptional neuroimaging research that's occurred that's really led us to identify a core neural circuit that is abnormal in OCD. And I've just highlighted two of sort of the seminal studies. These are both, as you can see, studies that happened 30, 35 years ago. The first of which on the left here is a study that was done by Baxter and colleagues using PET. So both of these are using PET imaging. And this study on the left is basically looking at abnormalities in brain metabolism between patients with OCD and normal control patients. And the finding back then was that there was hyperactivity in a couple core regions in OCD, namely these areas here in the orbital frontal cortex. And the other area that's really shown to be hyperactive are these areas here in an area of the brain, what we call the striatum, or more specifically the caudate nucleus. And so fast forward a few more years, these findings were actually shown to be replicated in patients with OCD and actually normalized with patients who took either SSRI antidepressants, which is the gold standard medication treatment for OCD, or we're doing behavioral treatment for OCD. Both approaches actually lessened this hyperactivity. And then another study by our very own Scott Rausch here at McLean Hospital was done a few years later, and he and his team took a little different approach. They actually looked at symptom provocation. So they had OCD patients that they put inside the PET scanner, and they actually had them do an exposure. So they identified something that each patient, would do to heighten their OCD symptoms. And they looked at which brain regions became more active. And lo and behold, they're very much the same as what was identified over here. Again, orbitofrontal cortex, the caudate, and another area of the striatum called the putamen. And then this other area, the anterior cingulate, which is yet another region that's been found time and time again in OCD to be hyperactive. So again, fast forward now decades later, these findings have been replicated over and over again in various types of imaging, whether it's structural imaging or functional neuroimaging, or even studies of the white matter in people with OCD. And so what this has led us to now is, as I said, this core circuit that's been sort of called, quote unquote, the OCD circuit, which involves primarily regions here in the frontal cortex, namely orbitofrontal cortex and anterior cingulate, like I mentioned, areas in the striatum, the caudate and putamen, and another area called the thalamus, which has formed this quote unquote, OCD loop, which has been really the focus of now more targeted treatments like ablative neurosurgery, or even more recently, deep brain stimulation to target several areas located within this circuit. And so this has all been fantastic, and it's advanced the field to some degree. But one thing we haven't found, despite all of these years of neuroimaging research in OCD, are what I would call definitive biomarkers, meaning a neuroimaging test or finding that could really guide clinical care. And so whether this is an imaging finding that helps us with diagnosis, so to diagnose OCD versus people who don't have OCD, or to guide treatment selection, even more importantly, what I term here is precision medicine, which would be basically a neuroimaging finding that could tell us this person would respond better to behavioral treatment than medications, or vice versa, would respond better to medicines versus behavioral treatment, or even beyond that, which medicine in particular would be most effective. So we don't really have any imaging findings that allow us to do that. Sadly, we don't have much of that in psychiatry in general, unfortunately. So what are the reasons behind that? And there are probably a number of reasons, but one is that OCD is a heterogeneous disorder. So when we think of OCD, we think of it as a single, disorder, one entity that is treated essentially the same way, with the same medications, the same behavioral treatment approaches. But in all honesty, OCD can include very, very different symptoms. So, classically, and what we'll talk about more today is contamination, so fears of germs or dirt and having to wash your hands over and over again. That's one of the sort of, more popular ones both in media and even clinically. But there are a variety of other things, like worries that somehow you're going to be negligent and make some sort of mistake or error and harm people that you care about, or symmetry and ordering, the need to have things be arranged in a certain way. And for some, this can involve what we call magical thinking, meaning that if things aren't arranged in a certain way, that something bad might happen to someone that you care about, for example. We can have checking behaviors where, again, this relates back to harm, where we worry about somebody getting harmed. Maybe we've left the stove on when we left the house. And so people with really severe responsibility for harm and checking will have to go back time and time and time again and check the stove, for example, check the doors, check the water faucets, you name it, sometimes to the point where they just can't leave the house. Worries about contamination not involving germs and dirt, so radioactivity, environmental contaminants, things like that. People can have what we call scrupulosity, worries about moral issues, that they are a bad person, that they've done something bad, or various other themes on that. Worries that, there are good and bad numbers, so they might have to do things a certain number of times, and if you don't, then something bad might happen. The list goes on and on. So I could spend through, the talk just talking about the various themes of OCD. But the point here is that we can put two people with OCD in the same room, and while they might both have obsessions and compulsions, the actual types of symptoms can vary dramatically. And so you'd think, well, people probably have looked at this when they've done imaging, and the surprising thing is that it largely has not been looked at. So we have many neuroimaging studies in OCD that have been done. I quote 120 here, but there's probably even more since I created this slide. But in my last check, a very small fraction of those actually looked at abnormalities in the brain that are dimension specific, so related to a particular type of OCD symptom. And even those studies that have done this, there's a lot of limitations. Many of them were very small, so they only included about 20 or less subjects, which can be problematic in terms of identifying abnormalities in such a small sample. A lot of them just looked at washers and checkers when, as I just mentioned, there are lots of other things out there besides just washing and checking. One of the biggest issues is that these studies largely use what I would call a categorical approach. So it's exactly what I talk about here, is that they basically split people up into being either a washer or a checker, when in all honesty, most people with OCD have multiple types of symptoms. So it's not being one or the other, it's usually a number of different things. And the better approach really is to use a dimensional approach where you are examining abnormalities or changes in the brain that correlate to different symptom subtypes in the same subjects. And so I'll talk about this more because this is the approach that we took in our preliminary study. And the last thing here I'll just mention, because I'm going to talk a bit more about it, is that all of these studies use what we call a group level analysis, meaning that they employed a technique which has pretty much been the standard in neuroimaging, where they use a population-based brain template or atlas to be able to do their neuroimaging analyses. And this actually creates problems, and I'm going to go into this in more detail in a second. So I wanted to give a little shout out here to our program here at McLean called the Obsessive-Compulsive Disorder Institute, or OCDI. And we've been around for, gosh, I should know this, but I think we're about around our 30th year at this point. Started by my mentor, Michael Janicki, who really is, one of the leaders in OCD treatment and research. And so in our program here is a residential program, so we have patients that come from all over the country to receive intensive behavioral treatment, largely for their OCD. And so it provides us an amazing resource for research purposes. And there are some advantages and honestly some disadvantages to doing neuroimaging research here. Many, if not all, of our patients come here with really severe OCD. One of the really, particularly valuable things about the OCDI is addressing what I just mentioned. It's an extremely diverse OCD population, meaning that we have patients who experience all of the things that I mentioned to you in the previous slide in terms of symptoms, and many others. And so it covers pretty much all of the sort of widely accepted symptom subtypes that I described. Another advantage is that we have people here for up to 3 months sometimes, which allows us to do longitudinal neuroimaging, which is extremely hard to do. If you could imagine, trying to have individuals who have a disorder that you can scan at multiple time points over the course of a treatment. It's rare to be able to do that, and we can do it here at the OCDI. And then in general, the folks who are here with us really are interested in altruism and wanting to advance the field and learn more about OCD. So there's a lot of advantages. There are disadvantages. The OCD population that we see here is typically very heterogeneous, not just in terms of OCD, but having other comorbid disorders like mood disorders and anxiety disorders and other things. So it does make it a little tricky to do very specific analyses when we do neuroimaging work. And there's also a level of unexpectedness, which is largely due to insurances, because while a lot of insurances do cover people to be with us, they will often decide at whatever time point that they no longer need to be with us and will no longer cover the treatment. And so people end up leaving sometimes abruptly or unexpectedly, which can make it a little more difficult to do imaging studies here. So because we have access to our population here to do imaging, we've been very interested in trying to do neuroimaging analyses to understand some of the things that I've already mentioned to you. And one neuroimaging approach that we've been particularly interested in using is something called resting state functional connectivity MRI, which basically is like getting a regular old MRI scan that you would get to look at the structural organization of your brain or the anatomy of your brain. But in this case, we're actually looking at levels of oxygenation in the brain, which is something that can allow us to actually look at the functional capacity and organization of the brain. And what's interesting about resting state functional connectivity MRI is that it takes advantage of the fact that for all of us, our brains are active all the time, even if we're sleeping or at rest and not thinking or doing anything. And we can take advantage of this by looking at these resting state, activity and looking at correlations between brain regions at rest. And what we find is that there are certain brain regions that seem to be more connected or less connected with other regions, such that they actually form networks or collections of brain regions. And you can see that a little bit here in this figure, where you see these yellow and orange regions, which make up essentially a collection of brain networks that are active at rest, what we call the default mode network. And so all of these regions, whether it's here or here or here, are all connected, or their signals are correlated in some way, and they're active at rest. Whereas these areas in blue, essentially here, are what we call task-positive regions, or regions that are active when your brain is actively doing something, and they actually turn off or become less active when you're at rest. And so we can identify these collections or networks of brain regions using functional connectivity MRI. And the great thing is it can be done at rest. So it's very, typically not too onerous for people, and the scans are relatively short. So we can put people in and get a decent resting state MRI scan, certainly within an hour, and oftentimes within 30 minutes or so. And so if you take that idea and go even further with it, you can use functional connectivity, resting state connectivity analyses to actually do what we call a parcellation of the entire cortex. So you're essentially looking at how each dot here, or vertex, is connected to the entire rest of the brain. Oops. And then go on and do that for every single spot here in the brain. And what you end up here with are these cortical parcellations, where you find various different networks or collections of brain regions that seem to be communicating more efficiently with one another than with other regions. So again, you can see this network in yellow here and here, which is akin to the network I showed you before, which is active at rest, or what we call the default mode network. But there are various other networks, all color-coded. And what we found is that there are functional capabilities of all of these networks that have been identified over time, that they all are involved in different sort of cognitive processes. For example, these blue and green networks here are what we call the motor network. So they're mostly brain regions that are involved in our bodies moving and doing motor exercises. So that's an intro to functional connectivity analysis. I wanted to get back to one thing that I mentioned before, which is this downside to using group level analyses. So this is what I was mentioning before, is that traditionally, when we've done these analyses, we've used a brain, sort of an average brain, for lack of a better term, to be able to do these analyses. And I'm sure you're all thinking, well, there is no such thing as an average brain. You know, all of our brains are different. They have different functional and anatomical organizations. And so there's something sort of innately wrong with doing that. But it's, you know, it's sort of been the traditional approach up until recently. And as I said, it's problematic because there's a lot of variability in how our brains are organized. And so without addressing this in the analysis, it's quite possible that you know, biologically important differences in the brain have been obscured because we just aren't getting sort of a clear snapshot when we use this group level approach. And so I was very lucky a few years ago to be introduced to Hisheng Liu, who at the time was downtown at our sister organization at Mass General, and had devised at the time a way to actually do individual level analyses. So this is a way to actually get around the problem that I was describing to you. And I'll turn your attention away from this extremely complicated sort of diagram of how this process actually works, and direct you more over to this right side here, which is just basically giving you an example of what happens when you apply this individual level approach. And this is basically taking three individuals that Hisheng and his group scanned over the course of 5 days. And the first thing you'll notice is that it's a very stable approach. So day 1 is no different than day 2, day 3, day 4, day 5, which is good. That's what you want to see, that you're not seeing a lot of variability with each scanning. If you look more closely, and it's not readily apparent when you just look at these, but if you look closely, you'll notice that there are subtle and even not so subtle differences in the organizations of these networks between subject 1, subject 2, and subject 3. So if you pull out any of these, for example, you know, and look at them, you can see that, you know, these are not identical. And it's support of what I was just saying, is that people's brains are not functionally organized exactly in the same way. And with this approach, we can actually adjust for that in the analyses, making it a much more precise analytical technique. And I really liken it to, you know, a blurry image, which is sort of the way neuroimaging analyses were done for decades, where we're using this group level approach and obscuring or blurring potentially important things. And now with an individual level approach, we get a much more refined and precise picture, like this picture of Rosie, one of my border collies. I'll show you my other border collie later in the talk. So what we did was use functional neuroimaging, resting state functional connectivity imaging, this individual level approach, and a measure of dimensional OCD symptoms. So this scale, there's a few of them. We used this scale, what's called the DOCS, which stands for Dimensional Obsessive-Compulsive Scale. And essentially what it does is it asks a number of questions about the severity of symptoms, but breaks it down, not just looking at OCD in general, but breaking it down into 4 distinct categories. You know, things that I mentioned to you before. So a category about germs and contamination, a category about being responsible for harm or injury, another category about unacceptable thoughts, like having unpleasant thoughts about sex or harm or violence. And then a last category, which are things relating to symmetry, completeness, or the need for things to be just right, which is in and of itself, you know, as you look at these things, you could say these are really very, very different things that people with OCD can experience. So our goal was to collect neuroimaging data, use an individual level analysis, like I mentioned, and used the DOX scale that I just showed you to be able to identify brain regions that were predictive of symptom severity within each of these symptom categories, the 4 that I just showed you. And we were successful in doing that in 2 of the 4. And so I show you those 2 here. We found a set of brain connections that predicted contamination and washing symptoms, the first category, DOX1. We also found another set, and if you look at these, it's quite striking that they're very different, really entirely non-overlapping, that predicted symptoms in the harm checking category. And amazingly, we were unable to find any reliable model or bunch of connections that predicted either DOX3 or DOX4, so the 2 other categories that I mentioned to you. And it's hard to know why. And the speculation here is that these 2 categories, I mean, when you see contamination washing, it's pretty distinct. There's really no question about it. It really is a fairly homogeneous group of patients. Same can be said for harm and checking. But the other 2 categories that I mentioned to you, I'll just go back to them briefly. Unacceptable thoughts, and even more so this category, which I call grab bag of symptoms, symmetry, completeness, not just right experiences. It really is still very, very heterogeneous, and it's quite possible that it's heterogeneous so much so that we're just not identifying brain regions that are predictive of it. But these 2 we were able to find, and so this represents not only the brain regions, but it breaks it down into the various networks. So again, these collections of brain regions that are color-coded up here. And what's interesting about this, I know these are abbreviated, and you likely do not know what these abbreviations stand for, but I will tell you that this green network and this purple network are both networks that are involved in attention, the dorsal attention network and the ventral attention network, and are not networks or even brain regions that were typically thought about in OCD before. And I'll even point out that, like, going back to that core circuit that I mentioned to you way back in the beginning of the talk, we don't see any evidence of that, for example, in the orbitofrontal cortex, which is a region that's always been implicated in OCD. When we look specifically at contamination washing, we don't see it here. So it raised a lot of interesting questions for us. And we began to think about, was there anything about these regions that sort of, mapping backwards made any sense in terms of contamination washing symptoms? And so we looked at, so this is essentially taking the 13 connections that you see here and just listing them out. And what we found was that there was a region in the right medial frontal gyrus, and I'm going to square it out here, which was particularly represented. And so what this means is that these connections, when they were stronger or more increased, the person had more likelihood of having worse contamination symptoms. There was one other region, again, looking at this, what we call right medial frontal gyrus, that's just the area of the brain that it's in, that was also represented over here, meaning that when these regions were less connected, they were predictive of higher contamination symptoms. But we were interested in these connections in particular. And when we mapped these out, the nice thing that we had, so I failed to mention that we scanned people both when they first came into our program, but we also scanned them when they were leaving our program, oftentimes, weeks or months later. And so we actually could look to see, were there changes in these regions that actually correlated with symptom improvement? And indeed there were. So as you can see here, as these regions became less connected, people's OCD got better. And this is represented here, where pre-treatment, the connectivity between these regions was higher and significantly reduced when they left treatment. And this was associated with OCD improvement. So again, even strengthens the argument that these regions in particular were involved in contamination. And when we pulled these regions out and just thought about them a little bit more and went through the literature, we found that they actually overlapped quite impressively with a network called the ventral attention network, which I mentioned to you a little bit. So this is from an entirely different study that was actually trying to map out these attentional networks. And you can see up here that, you know, these areas in red are very much overlapping with these areas in red down here. So we started wondering, well, what is it with the ventral attention network that actually could be playing a role in contamination? And it's worth giving a little background on the attention networks in the brain. So there's really two brain networks or collections of brain regions that govern attention to external stimuli. One is what we call the dorsal attention network, which are these regions in blue. And they're really important for what I call what's largely called goal-driven attention, meaning voluntary maintenance of attention in the service of achieving goals. So mainly these would be brain regions that are turned on when you're reading, doing a task, doing anything that's sort of focused attention, where you are trying to achieve a goal, accomplish something. These are the brain regions that will activate. The other region, the other network is what's called the ventral attention network, these areas in red. And again, the areas that are overlapping with the regions we saw in contamination. And this is really what's typically involved in what's called stimulus-driven attention. So it's really important for detecting unexpected but relevant stimuli in our environment. So, you know, we could be going along and doing something, looking at a computer screen, reading something, and if something was to pop up in our peripheral vision or in our environment that was particularly salient, our ventral attention network would reorient our attention towards that stimulus. And so that's the goal, and there's really a back and forth between these, as you can imagine, that if one was particularly active over another or vice versa, there obviously could be problems in our abilities to be able to focus and attend to things, and other things as well. So this is Scout, who's our other border collie, and I use her as a really good example of the ventral attention network because I would say that she has a very hyperactive ventral attention network. So this is Scout running towards us as we're calling her name, which looks really good, and it looks like, oh, she's going to come right back to us. But invariably, that doesn't happen because Scout's ventral attention network, she'll be employing her dorsal attention network and be very focused on us and running towards us. And then her ventral attention network will immediately reorient her attention to various other things that are going on out here in the periphery, usually four-legged critters that are running around, whether it be squirrels, rabbits, or other things. So her ventral attention network will cause her to reorient numerous times because she's seeing something out here rather than being able to really focus on the goal at hand, which is running to us for the treat that we're holding. So it's just an example of sort of, if the ventral attention network is to go awry, it really could create havoc in one's ability to actually be able to function in a normal way. And so as we thought about this more and putting all this together, we kind of realized that, hey, maybe this actually makes sense in contamination-based OCD. As you can imagine, people with contamination-based OCD are completely flooded and reoriented by feeling like things around them and their surroundings or their environment are contaminated in some way. When really, they likely are not, but they're just so overwhelmed and sort of scanning their environment or feeling like there are things that contaminate that it involves pretty much every second of their day. So when we look at these regions, we came up with sort of a model. So this is sort of a standard model, again, where these attentional networks, the dorsal in blue, the ventral in orange, are sort of interacting in a normal way, where the ventral attention network will reorient your attention to things, but that there is a filtering, there is a top-down filtering, as I would say, preventing your ventral attention network from being too active, which we don't want, obviously. And so when that's working, it kind of puts a brake on it. It doesn't allow you to be distracted by things that you shouldn't be distracted by. And perhaps someone with contamination OCD, that brake is not there, which runs through this brain region called the right medial frontal gyrus, which is, in fact, as you can see up here, the region that we found to be strongly implicated in contamination-based OCD. So if this brake, this filtering brake is not there, it can allow the ventral attention network to run rampant so that anything and everything is thought to be a contaminated stimuli, and this cycle just loops and loops and loops without ever being stopped or filtered out. And so this was our working model, and we thought, well, how can we test this and potentially normalize it in some way? And one way to do that is to use transcranial magnetic stimulation, or TMS, which I imagine a lot of you have heard about. And essentially what that involves is having a coil which can create a magnetic field which passes into the brain tissue and can result in changes in brain activity. And when you do it repetitively, the changes in the brain are more long-lasting, and the function itself can be inactive. And so, as many of you are probably aware, there are existing TMS treatment protocols for depression and OCD that exist. I will say that the OCD protocol, just like all the treatments we have in OCD, is symptom non-specific. So we use it regardless of the symptom subtype. In our case, again, as I go on and tell you about this, the TMS treatment we're using is actually specific, we think, or hope, will be specifically for contamination symptoms. So one limitation of existing TMS is that it's just not very accurate or specific in the targeting. For those who have had TMS or those who have not, the way TMS typically is targeted is by using landmarks on the head to be able to find the target region in the brain that we're stimulating, which, again, getting back to just the pure diversity in the organization of the brain, makes one think, well, that just seems like not a very exact process, and it's largely not. But it can be improved by using a technique called neuronavigation, which actually uses each individual person's either structural scan, or in our case, we're actually using the resting state scans that we collect in our study to identify the brain coordinates to be used for TMS targeting, and then using this elaborate infrared system to actually use the scan to find the cortical landmark on the head to be able to do the targeting. And so in our TMS study, which I'm going to tell you a little bit more about in a second, we're actually using this approach to target the right medial frontal gyrus. Again, that reason that I mentioned to you that was so strongly implicated in our findings in each of our participants. And again, really to find the region within that area that's strongly connected to this network of regions called the ventral attention network. This is just a little schematic of the study that we are just getting off the ground now. And so we're bringing in, or hoping to bring in, around 50 individuals with OCD to collect a scan at baseline, and to do resting state connectivity, again, with the plan to identify in each person their unique location of their ventral attention network and the area in the right medial frontal gyrus that we want to target in that individual person. So it's kind of represented here. And then once we do that, people get randomized to get potentially one of three different treatments. Two different types of TMS, where these are different protocols of TMS, and we're using both because we're honestly not sure which of these is going to accomplish our goal. So we decided to have an arm for each of these so that we'll have individuals who are receiving both. And then people can also be randomized to a sham condition, meaning they're not getting the active treatment, which is really critical in a study like this. to be able to make sure that the findings that you're seeing are unique to the active treatment, not due to some placebo effect. And so we will end up treating people with our one of our 3 treatments. We'll end up scanning them again after their first treatment, then after the 10th treatment, and then after the last treatment. So they'll have a total of 30 treatments over the course of 6 weeks. And we can, with these scans, we can look at a couple different things. The main thing we'll look at is again, functional connectivity to see if there are changes in the connectivity of those brain regions making up the ventral attention network, which is really our primary interest in this study. We're also collecting task-based functional imaging using a task, and I'm going to show you this briefly. But this is basically a version of a task that was developed by a different group to look at the ventral attention network. So it's a probe, essentially, of the ventral attention network. And what it involves, the version we're using that we developed for this study, shows 3 different images. One is what's called a target image, and the 2 others are distractors. In some cases, there can be 3 distractors and no target. But people are told what the target image is. So for example, in this slide, this contamination-based image is the target. These 2 are distractors. And people are told, while they're inside the scanner, so this is being done in the scanner, to press a button when they see the target. And so there are various combinations of images that will flash up on the screen relatively quickly. And each time it flashes up, if you see the target, you press the button. And the types of pictures that people will see will vary. There's essentially going to be 3 different combinations. One, like this one, where you have a contamination target and what we call 2 neutral distractors, meaning these are non-contamination-based images. You can have a neutral target and 2 contamination-based images, or you can have all neutral. And so people will do this task. And what it allows us to do is actually probe the ventral attention network. So you can see that a little bit here. These areas in blue are all regions that are in the ventral attention network and that are actually being turned down, because what we're looking at here are what areas are on when people are just looking, what we call searching. And that's really when the other attention network, the dorsal attention network, is firing. So the people are looking at the screen, searching for their target image. And when they see the target, that is honestly when the ventral attention network should be activating, which we don't see here. So that's a little bit about the task. So we're actually going to be looking at differences in that as well. And as I said, the main aim of the study really is to look at changes in connectivity so that we can show with this treatment, the TMS treatment, that we're actually changing the connectivity between regions within the ventral attention network. And we'd see this either with the active treatments, but not with the sham. That would be the ideal. And our secondary aims really is to look at these changes in the task that I just showed you. And lastly, while it might seem most important to all of you out there, we will be looking at changes clinically. But I will say that for this study, we are really primarily interested in looking to see that we are changing the neurobiology of the brain, and the clinical results of that are sort of secondary. If we're able to confirm that with the treatment, we're actually changing the connectivity in the regions that we hope to, that will then allow us in our grant, which is through NIMH, to proceed to a larger study, which actually will be much more focused on looking at the clinical effects of this treatment. So hopefully, we get to that point and can show that the treatment itself can reduce contamination and washing symptoms of people with OCD. So that's essentially it. There are a lot of people I just want to thank briefly that were involved with this work, a number of whom are here at McLean, many of whom are in the OCDI with me. I mentioned Mike Janicki and Scott Rausch, who are really my 2 primary mentors with a lot of this work. Also, my home base here at McLean is in the Biological Psychiatry Laboratory when I'm not working in the OCD Institute. And my collaborators here, Jim Hudson and Skip Pope, have been instrumental in helping me with this work as well, along with numerous research assistants. Currently, Rosie Sokol is my working with me, and it's been great in getting the study up and running and actually beginning to enroll participants. And then there are a number of other people here at McLean who are assisting me in various parts of the study. Dan Dillon with the fMRI task, Justin Baker with a lot of the connectivity work, Mark Halko now with the, helping me with a lot of the TMS-based work. Chris Perriello and Gianna Schnabel are two past research assistants who have now moved on to graduate school. And then a few people down at MGH who have been helping with the functional connectivity work, along with the Shing Lu, who I mentioned before, who's now down at the Medical University of South Carolina. And inordinate amounts of funding that have also been helpful in allowing us to do this work and the work that is to come. So thanks so much. I'm glad we got through a lot of that, and actually it looks like we have a good amount of time for questions as well.
Absolutely. Brian, thank you for an excellent presentation and the work that you're doing on this important topic. We have a number of questions about aspects of OCD. One question that a few people ask relates to OCD and hoarding issues. And I'd like you to please say a little bit about OCD and hoarding.
Yeah. So the relationship between OCD and hoarding has been developing, I would say, over the last few decades. It wasn't that long ago that hoarding was really thought to be one of these subtypes of OCD. So when we used to do dimensional measures of OCD, like I showed you the docs, for example, they usually would have a category that would include hoarding. So not only would we ask about contamination or responsibility for harm, we would ask about hoarding as an OCD symptom. And it was largely thought to be, you know, a type of OCD. However, more recently, and with the advent of DSM-5, which I guess wasn't that recent at this point, but when DSM-5 came out, the feeling was that we had enough evidence to suggest that hoarding actually probably was really its own distinct disorder that was related to OCD, but there were enough differences, both in terms of the biology and the treatment, that it really warranted having its own, its own diagnosis, diagnosis or disorder category. And so we really don't think of hoarding disorder as being an OCD symptom subtype anymore. We really think of it as a standalone disorder. It still is within what we call the OCD and related disorders category, which was created by DSM-5. But as I said, the differences are enough to say that this is not related enough to OCD to call it, to put it under that umbrella. And it's namely because, the biology, so at least what we know about it, the little that we know, unfortunately, about hoarding from neuroimaging is that some of the core structures are the same as OCD, but there are differences as well. And even more importantly, that the treatment is different. So, I mean, it's related, but, typically, as I mentioned, I think way in the beginning of this talk, I mean, the gold standard medicines for OCD are typically the serotonergic antidepressants, the SSRIs, and an old tricyclic antidepressant called clomipramine. And those medicines get used in hoarding, but in all honesty, they're not nearly as effective, and there aren't nearly enough research to actually really definitively look at them in hoarding. So while they get used, we don't really have gold standard medicines that are specifically designed and developed and studied in hoarding. And so it really is, I hate to say it, but it's one of those things that's sort of an orphan disorder that just hasn't been studied enough to look at what treatments are most effective. And that can be said for the biology as well. While there are studies out there looking at neuroimaging and neurobiology of hoarding, there's not nearly enough. So that's my little bit on hoarding. I would say that with hoarding, and this honestly could be said for OCD as well, really the most effective treatment we have is behavioral treatment. And in hoarding, it's a very specific type of behavioral treatment, which is essentially dehoarding, which is an exposure in and of itself. It really uses the exposure based treatment property of sort of exposing someone to something that is incredibly anxiety provoking. And so for people with hoarding, that's essentially forced decluttering or dehoarding or getting rid of things, and doing it with someone who is trained to do this and to work with someone to manage it, with the hope that over time it might get easier and they may habituate to the anxiety that's associated with it.
Good. Thank you. Very good explanation of the differences and the information that we currently have on that. You mentioned that people to McLean for treatment, more severe and significant OCD. I would assume that means that they've had various types of treatment already before getting there. What do you do that's different to help people who may not have responded to other treatments in the past? What do you do for these people that are coming to McLean?
Yeah, that's a great question. So absolutely correct. The vast majority, if not all, honestly, of people that we see in our program are people who have traditionally failed or not been too successful with standard treatments. And by standard treatments, so medications, again, the usual gold standard OCD medicines like the SSRIs and other things. And then most, if not all, the people that we see here have done at least some outpatient exposure-based treatment. But the key here is that it's outpatient, meaning that it's your standard, maybe once or twice weekly, 1 hour exposure session with your outpatient therapist, and then you're left sort of out there on your own to try to do that without any coaching or guidance from a therapist. And what we do that's so different is taking that exposure-based treatment and doing it in an extremely intensive and supportive way, so that people here in our program will do literally hours a day of exposure-based exposure and response prevention therapy, the gold standard behavioral treatment for OCD. And some of that will be even with a coach. So we have coaches that are here essentially 24/7, but more so during regular business hours, that will actually coach people through these exposures on a daily basis. And then some of it will be non-coached, meaning that you're doing it on your own. But the point here is that it's taking traditional outpatient exposure-based treatment and doing it in a much, much more intensive and robust way. And that is really what the difference is. And that is what oftentimes gets people better while they're here. We do, various other types of treatments. We do, people will see psychiatrists while they're here, and we'll have medication changes. And we do various types of group treatments, that are largely based on your typical, your standard symptom subtypes. And we do family treatment as well, which is actually also very important. But the thing that really gets people better is doing intensive exposure-based treatment.
Thank you. There's been a lot in the press about psychedelics and a variety of conditions. Is there any work being done on the use of psychedelics potentially for the treatment of OCD?
Yeah, also a great question. So yes, I wish I could speak to sort of details on this. I'm not closely involved with some of that work that's happening, but I do know that it is happening. I think, not knowing this literature at large very well, but I think some of that work is looking at psychedelics as sort of an augmenter or enhancer of psychotherapeutic approaches. That may be happening in OCD, but I think a lot of the work is actually being done just looking at it as a standalone treatment. And I think psilocybin really is the big thing is currently being looked at for OCD treatment. And I believe there have been some preliminary studies and maybe even a larger study that may even be happening actively or is soon to happen, looking at psilocybin in OCD. And I believe a lot of that work is happening at Yale.
Okay, thank you. Often people with OCD may not seek treatment. They may be embarrassed. They may people around them may or may not even realize what they're going through. Could you say a little bit about if somebody out there is listening and they are experiencing symptoms of OCD, what do you say to them to encourage them to seek help?
Yeah, so I mean, that's a great question, man, and a question that, I think could be applied to various other disorders, not just OCD. I mean, I think, obviously a lot of this revolves around stigma that is just unfortunately still out there, revolving around mental illness in general. In OCD, I mean, I think sometimes what over and above that, what holds people back may honestly be concerns about OCD-related concerns in general, which are, some people may be fearful for an OCD reason to come into treatment. Unfortunately, while there's a bright side and there's an upside and a downside, to exposure-based treatment, it's an extremely, extremely effective treatment. Better than any medication that I could prescribe to anyone, but it is really challenging. What exposure and response prevention treatment involves is essentially what it sounds like. You're exposing people to their obsessive fears, to things that they worry about, and then preventing them from going through the compulsions and rituals that they will typically do to relieve that anxiety. So it can be really challenging, and oftentimes people can be really quite fearful of engaging in it. And all I could say in that regard is that it really is by far the most effective treatment we have. And I would argue, one of the most effective treatments we have in psychiatry in general, if people are really willing to put themselves out there and do the treatment as recommended by their, by their therapists. So it's really an argument to say this is worth it. That if you want to get your life back, no time is better than the present to get in the hands of a really good and skilled behavioral therapist and start doing some exposure-based treatment, because if it hasn't been done, if someone hasn't done it, they're really potentially missing out on something that could really change their lives.
Very good guidance, that even though it may be difficult, and anticipating it may be even more difficult, it really can help and make a big difference.
I've seen. I'll just say that I've seen that time and time again, which is why I feel like I can say it with so much confidence, that if people are willing to do the treatment, that it really can be helpful.
Good, wonderful. It's good to say that so people could hear it, and then hopefully make a decision to seek help. Brian, I want to thank you for the work you're doing, for just a wonderful presentation today, and for the work that you're going to do moving forward. I also want to thank everybody who joined us today, and I'd like to ask people to consider making a donation to BBRF. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientist. To make a gift, please visit our website, bbrfoundation.org, or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with family and friends, please visit the events and webinars page on our website. And I hope you'll join us again next month on October 11th at 2 p.m. Eastern Time, when Dr. Sahib Khalsa, Director of Clinical Operations at the Laureate Institute for Brain Research and Associate Professor at the Oxley College of Health Sciences at University of Tulsa, will present Exploring New Pathways of Discovery for Eating Disorders. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you. Take care.